Good morning. It's very early in the morning for me still, but I had coffee, which I usually don't drink, so hopefully that will get me up in addition to tea. I want to thank Peter for that talk, really talking about the environment, and now I'm going to switch a little bit, talk about genetics, but I think as we all know, it's a combination of genes and the environment that leads to these disorders, very complex disorders. And because my talk was switched, I was originally a full omics talk without a ton of genetics. I was asked to put a little bit of genetics into, so I have a few extra slides, so forgive me, but I was asked to, so that it could help frame some of the discussions that are going to follow after this. So let me see if I can walk and chew gum. I also just wanted to thank the organizers and also congratulate them. There aren't enough meetings like this where we bring basic developmental neurobiology with clinical focus, both looking at normal and disease or disorders, and I think this kind of meeting is essential and more of them. Let me see if I can do this. Does that, let's see, forward. I'll try this. There we go. Whoops. So here's my talk, and so conflicts of interest. I'm on some scientific advisory boards. I'm not going to be talking about work directly related to this, but I'd like to make those available. Before I even start, because I sometimes have a tendency to run over, I wanted to thank the people who really did the work, the people in my lab. It's a long-standing work from Neil Parikshak. The people with the stars have already left, but Neil Parikshak, Mike Gandel, Jason Stein, Hae-Jung Won, Luis de la Torre Ubieda have driven a lot of the work that's published, some in collaboration with Steve Horvath and Giovanni Coppola, who are long-standing collaborators. And I'm going to present some work from collaborators in iPsych as well as my collaborator Sergio Pasca at Stanford, who is a pioneer in the kind of growing of stem cell organoids. So, genetics provides a simple paradigm in a way, and that's kind of a joke. We think about, we identify things that are heritable, we find genes, or we find loci. Even finding the loci, then one has to go and find the gene, because often the loci, coding genes are only about 4% of the genome. 95% or more is non-coding and usually poorly annotated, so we have to then take the SNPs, if we're doing a GWAS or whatever the variant is. Most genetic variation that predisposes to disease lies outside of genes, and so we have to find the locus, then the gene, then figure out what it does, which is no mean feat. But the point I want to make is that these are highly prevalent disorders, from kind of Tourette syndrome, schizophrenia, autism at the bottom, around the 1 to 2% range, to 20 to 40% for things like anxiety, depression, et cetera. And the heritability is very high and variable for all of these, but it leaves substantial room still for the environment. Again, this is measured heritability depending on twin and family-based studies, and then SNP heritability, which is limited by the size of the GWAS as well as the methods that are used. But one can see the SNP-based heritability is the proportion of the heritability that is attributable right now to measured common variation. So there's room for both common and rare variation in these disorders. So again, the reason why genetics is so interesting in a way is because it is a finite space, and when one has well-powered, well-designed studies, you can find loci and you can find genes and determine how they cause disease, and that's essentially what we try to do. Of course, I could say in a kind of ironic way that when I started working in this area, I'm trained as a developmental neurobiologist originally. When I started about 20 years ago, my thought was we'd find a handful of genes that would contribute to these disorders, and then maybe that would take 20 or 30 years, and then we'd have a little bit of time to work on them, but it's, the genetics has moved very rapidly, and so that's caused a number of issues that I'll discuss in a moment. One of the things that Peter mentioned is the overlapping heritability, and I was involved in that brainstorm, but I want to make a slightly different point. This is the genetic correlation between bipolar schizophrenia and autism schizophrenia. There is a correlation, it's 0.2, but it means that most of the risk for autism is distinct from the risk from schizophrenia, but surprisingly, they share a substantial and highly significant proportion but far less so than bipolar and schizophrenia. But there is this conundrum, and in a way the genetics is going to perhaps allow us to help pull apart these shared factors and how genetic predisposition may interact with the same environmental stress to kind of lead to one or other of the disorders or increase risk. So the way that I'm going to frame this today is that, focusing on autism, genetic risk for autism is complex. It involves many loci, but there actually is evidence for convergence from many studies, and I'll present that both in terms of timing, cell type, and biological pathways. And then I'm going to kind of present a kind of a framework or model where I'm going to say that understanding the mechanisms through which genetic risk factors act necessitates having maps. And we're kind of in a map building phase. Maps of gene regulation in developing human brain, normal maps, normal gene regulation. And so we started to do this about 6 or 7 years ago, starting with EQTL and 3D chromatin maps, as well as using ATAC-seq for open chromatin, and now more recently single-cell transcriptomes. I'm not going to have time in this short talk to talk about our work in single-cell transcriptomes, but I can refer you to a paper on bioRxiv that has 40,000 cells from developing human brain, and we make the data available. But these maps, and if this is all you get out of this, totally fine, because this is all really I'm saying, implicate early cortical development as a substantial period of risk for both early and later onset disorders. It fits with the environmental work very cleanly, and whether it's autism, which is a childhood or very early onset disorder, or major depression or schizophrenia, early neurogenesis, essential period of risk. So autism, now to autism. It's a heterogeneous neurodevelopmental syndrome. It's not defined by etiology, so it should be no surprise that it is not only clinically heterogeneous, but genetically heterogeneous, and I want to make a couple very quick points. Autism is now defined by, let's say, problems or abnormalities in social cognition and mental flexibility, differences in these areas compared to neurotypical children. But about 10-15% have epilepsy, 30% have intellectual disability. Again, it depends on the cohort. So there's this overlap, clinical overlap, and this is just examples. There's a clinical overlap with a lot of disorders, including ADHD. You might have, you could co-diagnose in about 40% of patients. And this has kind of been our model for a long time. I'm not going to focus on this aspect of the model, that you have kind of a bunch of risk factors here in red, environmental being a very large one, but also large effect mutations, and then the small effect common SNPs that in an individual come together to tip the balance towards a disorder like autism. They increase risk. They do not cause autism. In fact, there aren't autism genes. These are genes that disrupt neurodevelopment and then increase risk for a panoply of disorders. And some genes have more specific effects than others, and we're just beginning to ferret that out. But the notion was that really brought me into autism research was that it's part of a continuum of normal variation in a bunch of different dimensions: social cognition, language function, and repetitive restrictive behavior, shown here from a review in 2007 actually now in Cell, where the idea is there's a contour of normal distribution, and at some point genetic factors bring you, and environmental factors bring you over past the tipping point where society now diagnoses you with a disorder. But again, this is part of a normal distribution for the most part. Now, if you have a very large effect mutation like Fragile X, that's going to have a very large effect and kind of take you way out of the normal, kind of way into the tail. And so, again, I think the model here is kind of one that's I'm borrowing from Waddington's framing of the epigenetic landscape in the 60s as canalization. We have brain development occurring, and it occurs, we are relatively resilient to a wide variety of environmental and genetic factors that might knock the brain off normal development. And those with a large effect size mutation just can't respond appropriately. So development has been knocked off the track, so to say. I hope that makes sense. And then that increases risk, for the disorder. So again, this notion of not the genes are causing the autism, they cause abnormal development. Maybe they make the brain less able to adapt to some environmental or other stress, and then the child ends up having an autism spectrum disorder. And now we know that genetic testing can identify almost 20% of mutations that contribute to ASD if one does a full genetic panel. So from a clinical standpoint, if you do a clinical microarray, a bunch of single gene tests like Fragile X and exome sequencing, in our center we're getting about 15 to 20% of patients with a large effect size mutation. And what's interesting is that those mutations are themselves each very rare. This is showing a kind of strongly implicated de novo single nucleotide variants. These are kind of new syndromes caused by mutations that are being identified, but none of them are more than 1%. They're all rare. Here are the copy number variants, which are a large structural chromosomal variation that's either a deletion or duplication of genes, and then some of the known syndromes that have a large proportion, like Fragile X, at 20% of the children diagnosed with autism. These rare conditions right now account for, let's say, around 5, 6, 10% of autism. There's more to be discovered. And these are mostly de novo events, like Down syndrome. They're not inherited. Most of what's been identified so far, and I'm just going to briefly allude to the fact that some of that has to do with the study designs that have been done in the major sequencing studies have been done in the Simon Simplex collection, which is depleted for inherited risk. It's really focusing on identifying de novo mutations, and that's been the low-hanging fruit, and it's been very successful. So we have part of autism, maybe we estimate 15% or so, is going to be a collection of very rare disorders. Maybe on top of some of those rare, you'll have the polygenic risk, predicted common inheritance, and then there's unaccounted for risk, which could be just measurement issues. A lot of it could be common inherited, and some of it could be rare inherited variation as well. So just to summarize, it's predicted that there'll be many causes of autism that are rare genes, hundreds if not more than 1,000. None account for more than 1% of cases, highly additive effects, and very strong pleiotropy. So this is a challenge, obviously, to neurobiology and any mechanistic understanding. Worse than that is that we have to go now from genetic variation to connect all of these different hierarchies and levels to get to behavior and to get to understand the brain. And I'm going to focus today mostly on mRNA and protein levels, not because I feel that's the only place we should go, of course, but because it's tractable. I can profile RNA in large sample sizes, very reasonably priced, and I can detect almost every gene expressed with the very high dynamic range of these technologies. So we do this because it's a kind of readout of the genetic and environmental factors, but of course we're eventually interested in connecting all of this, which is an enormous challenge. And today I'm going to talk about the transcriptome at this angle. I've mentioned some of the genes, and I'll talk a little bit about this, knowing that the job is to kind of now start to connect these two, which I think is feasible. And the way that we connect, of course, different measurements are made at all of these different levels, so how are we going to connect them? One of the ways that we've been thinking about things for about 15 years is genes don't act alone, almost nothing in the brain. Brain regions don't act alone, they act in networks. And so we think about things as kind of coordinated networks. So I'll just give you a very quick cartoon. If we're looking at gene expression, which is the RNA expression, you have samples here. This is just a cartoon. And you can see that there are different genes, each with a different color here. And they show different patterns of expression, different patterns of covariance. And from that covariance, we infer co-regulation. We have to prove that, but we can infer that. And these groups that, let's say, go up and down together in this particular group, gene C, form modules. It's just a term. And within the modules, there are the central genes, which are hubs. And it turns out that these hubs are essentially, really represent the first principal component of that module, or the major function of that module. And so this is a cartoon showing that when we took whole cerebral cortex in like 2007 and 8. Mike Oldham, who was in the lab at the time, we took hundreds of samples from the Webb gene expression studies, and when we actually looked at what these modules were, we were able to do an in silico dissection identifying the various cell types of the brain. So by having whole tissue, you can pull out the major source of variation in the transcriptome, which was the cell type composition. And but you also get other features. You could, you get features corresponding to many different levels and elements of biology. This is just a cartoon. So we've used this approach to ask three major questions. I'm just going to frame this in that way. Can we use transcriptional networks? Of course, the answer to all these is yes. Can we use transcriptional networks to characterize the molecular pathology of a neuropsychiatric disorder, autism? Yes. Can we use it as a quantitative phenotype for cross-disorder comparison? So again, just like you might have a psychological test of social function or some adaptive function, how about the transcriptome as a quantitative molecular phenotype? And then, more importantly, can we use these networks to actually move to function? So I'm just going to summarize some published work, move on to some unpublished work. So despite etiologic heterogeneity, I told you have 100 cases of autism, you probably have 100 different causes. In this first very small study with microarrays published in 2011, led by Irina Buonagu, she showed that 2/3 of cases showed upregulation of microglia and astrocyte genes and downregulation of a set of neuronal genes involved in vesicle transport and synaptic signaling with a kind of RBFOX1, a neuronal specific transcription factor, as the hub. And the causal genes, in other words, when we looked at what was known at that time, which wasn't that much, where the causal genetic signal lied, in which process, it was the genes affecting the neuronal component predominantly. This was really a preliminary result. We've subsequently gone on to show, and I'll show you this in a bit, that microglia upregulation is not something that's seen broadly. It's fairly specific to autism. Then in 2016, we had a much larger study, 3 times as large. We're looking at cerebral cortex, temporal lobe and frontal lobe, and cerebellum. Very small signal in the cerebellum. Most of what I'm talking about is occurring in the cortex. And this study was able, with more samples, to refine the signal a little bit and identify these 6 shown here, 5 that correspond to cell types, and very strongly, this M20 not so strongly to cell types. 1 that's microglia, so we're able to separate out the microglia and astrocyte component, which are both upregulated, and separate out different components of the neuronal signature, so slightly refined. So, a lot of people said, this is just transcriptomics, it's BS, it's just like post-mortem artifact. And, we were able to kind of control for those confounders, but I can't help hearing what people say. So we've used a lot of methods now to look at this that are independent of RNA post-mortem factors. Of course, this is microRNA study, and it shows a very similar pattern. But the major point I want to bring up is that if we look at H3K27 acetylation, which we did with Shyam Prabhakar in this paper led by his son et al., we find that we get a similar pattern of H3K27. It's marking enhancers, putative enhancers. So we can separate the autism cases and controls, and we can show that this enhancer pattern actually underlies some of the abnormal gene expression. And again, we see in prefrontal cortex and temporal cortex a very highly overlapping pattern. So, the kind of technical artifacts that might occur with RNA sequencing won't occur with this. You could still come up with some other explanation. But we really felt very confident at this point that what we're looking at may not be causal, but it's a defined molecular pathology of a neuropsychiatric disorder. And the reason this is so important is because there's no, unlike Alzheimer's or Parkinson's, there's no kind of neuropathology of these disorders. And here, what we're saying is, yes, there is a neuropathology. We can define the neuropathology, but we have to use molecular techniques. We can't see it under a microscope with an antibody. So multiple techniques support a shared neuropathology. It's not in every patient. It's in about 2/3, let's say 65 to 70% of patients. So about 1/3 don't show that pattern. So again, the way that I think about this, again, this is my biomedical framing of this, just very broadly. Cancer is a disorder of proliferation and invasion that's caused by a combination of genetic susceptibilities and in some cases environmental contributions, of course. Asthma, also a gene environment. Hyperreactive airways would be one way of very concisely summarizing it. Stroke is a disorder of acute brain hypooxygenation, due again to many vascular and cardiac etiologies. In this context, what is autism? Obviously, I don't really know in this context yet, because in these other disorders, we have models of how those organs work, and so therefore we can put the pathophysiology within the context of that normal functioning. In the brain, we really don't, so I can just describe the neuropathology here. We have altered neuronal gene expression, which is downregulated, an increased kind of neuroinflammatory component. The causal genetic variation actually leads to alternative splicing defects as well as this transcript overexpression. So far, everything we've looked at, and I'm going to show you some more, shows that the genetic variation is actually sitting, the causal anchor, sitting in the neuronal part, but they're always associated with that and probably kind of reactive and part of a vicious cycle is this increased microglia and astrocyte gene expression. We don't know, early on it might be adaptive, but I would assume chronically it's probably maladaptive. So the major question that we were asking ourselves at this time is how specific is this to autism? So can we use these network-based framework as a way now to ask about disorders? And this was done, and it's beautifully by Mike Gandel, who was a postdoc in the lab. The notion is you have clinical syndromes, various etiologies, and that the altered brain transcriptome could be an intermediate phenotype. And again, from the PGC in 2013, we knew that there was these shared heritabilities shown there on the right. It's showing their high heritability, and then in the light green, the co-heritability of these disorders, which again I had summarized in the initial slide. So the cross-disorder analysis, without going into too much gory detail, we downloaded microarray studies in the first case, almost 900, and we ended up with 807 decent samples, and then it was a year of processing to prove to ourselves that we weren't, just filled with confounders, because these are different studies. So we did both differential gene expression and a network analysis. And I'll just show you some of that summarized. And this was published last year, I think in February. This is showing the correlation of gene expression between these disorders. The IBD correlation with autism is positive, but it's not significant. But it's due to an overlap of neuroinflammatory genes expressed in microglia with a lot of the inflammatory genes expressed by the innate immune system in IBD. Bipolar and schizophrenia have a positive and significant correlation with the autism transcriptome, but much more so with each other, and they have a weaker but significant correlation with major depression. So the question is, what is underlying this kind of correlation? Is it related to the same factors that lead to the genetic correlation? So we can basically ask that in a fairly clumsy way shown here, but I think it's fairly convincing. If we take the SNP-based correlation of these disorders from published PGC data, and we did new RNA sequencing on top of the microarray from a few of the disorders for which we had tissues from collaborators as part of the PsycENCODE consortium, we're able to show that there's a Spearman's rho of 0.7 between the correlation of two disorders' transcriptome and their genetic correlation. So there's about half the variance in these relationships can be explained by the genetics. So this isn't some kind of odd artifact. It's not that what we're seeing in the brain at the transcriptome is causal, but that it's at somewheres related to this initial kind of genetic etiology. When Mike did network analysis, he identified a bunch of different modules, and they group by cell class. So the glia down here on the left, the neurons. The red lines here, this is just a multi-dimensional scaling plot of the first two using the first two principal components. And you can see that the neurons are up there and the glia are down here. The red is a negative correlation. A green is a positive correlation. So there's this, same relationship we had kind of seen before in autism. But now we can ask which of these are related to which disorder and how are they going? And, in a broad way, we look at all the neuronal modules shown up there, the CD1, 13, 10, and 5. Those are all down. There's an astrocyte and microglia module, which are I guess what you call yellow and green yellow, which are up. But if you look at schizophrenia, it's only the yellow that's up, which is the astrocyte module, not the microglial module. And to a lesser extent, the neuronal modules are all similarly affected. And this is just showing alcohol abuse disorder in the middle, which is not showing any of those patterns. And major depression kind of has was very underpowered here. And again, didn't really overlap much with autism, slightly more with schizophrenia. So this, again, then we ask the question, where is the genetic, if we look at the polygenic risk, where is that lying? And it's lying in the neuronal modules. So what can we do with this? Well, we can begin to explore and ask, what do these things mean? And I'm going to skip over this relatively quickly just to show you that at a meso and microscale level, one of the modules, this purple module that's downregulated in autism and schizophrenia, is related to both brain activity measured with functional MRI as well as firing rate of neurons measured from a mouse study done by Sasha Nelson that then we reanalyze the data from. So I think one can begin to, as one has gene expression patterns from various developmental neurobiological studies and perturbation studies, to begin to understand what these are and begin to make the links between these patterns that are seen in postmortem brain and actual function. What function do they represent? Recently, we led the PsycENCODE study, which was a huge collaborative effort, but used RNA sequencing in over 2,000 control subjects, schizophrenia, bipolar, and autism, and essentially replicated what we had found before, but now refined it substantially, because instead of having under 10 modules associated with disease, we now had 60. And so we massively refined the transcriptional networks. And what we could find with the RNA sequencing was that isoforms show the much larger disease and cell type specificity than gene-level analysis. And so just to give you an idea, and I just refer you to the paper, because it's too much to go over in a short talk like this, but I just wanted to bring your attention to it. This is just a subset of the modules, each of them organized by those that represent different neuronal classes, excitatory or inhibitory, glial modules, and non-cell type specific modules, and their relationship to disease, red being upregulated, blue downregulated. And one can see this really has allowed us to substantially refine and identify much more disease-specific and disease-shared signals, which I'm happy to discuss in more detail at some other, in other time. But again, we can annotate these functionally, it's really a quite interesting set of patterns. So then the major question is, can we use these networks now to understand where disease risk genes are acting or to inform disease modeling? So we ask the question using networks derived from normal brain development. So now we have transcriptome from fetal brain up to about 3 years of age from the BrainSpan Consortium, and we ask if we create networks that correspond to the different cell types, epochs, stages, and processes, what do we find? So again, if we take the different classes of autism mutations, those large effect de novo with the large arrows, or the common variation down here that's having smaller effect sizes, where does it live? And so this, again, is within the context of normal neurotypical brain development. The genome, the transcriptome is organized into co-expressed modules, and then we can ask, is it distributed across all of these processes, or is it enriched in one? And indeed, we find that it's enriched in molecular pathways that are annotated for transcriptional regulation, chromatin modification, and neurogenesis. And I'm just going to, summarize this again, really remarkably. And when we look at genes that cause intellectual disability alone without autism, those do not show the same enrichment. So again, this is just a cartoon summarizing a lot of figures, but that the period where risk genes are most highly expressed is mostly in this embryonic period and just slightly postnatal. And we basically were able to divide it into two modules, or two processes. One that involved two modules involved in transcriptional regulation, which was up right at the peak of neurogenesis, and they cross around 16 weeks gestation when synaptic development starts. And then the common genetic variation is in synaptic development. And when we ask what cortical layers these genes might be enriched in, if any, we find mostly the superficial layers, layers 2 through 4, which are involved in cortical connectivity rather than cortical-fugal connectivity. So that's what we got from the when and where. And then this study was done in 2013 based on a limited amount of exome sequencing. Now there have been multiple studies, and we can say that the two developmental modules that were enriched, called M2 and M3, that were enriched in chromatin and transcriptional regulation during neurogenesis are indeed even more strongly now enriched in a number of different autism studies, both from Sanders, Iosifov, the DDD study, which is just broad developmental delay, and this IHEART study, which is multiplex inherited autism, which is not yet published, but which has been on bioRxiv. And just to show that, this has to do with inherited risk now. It's the first study because we use a different kind of ascertainment scheme. Rather than simplex families with one affected autism, we're looking at multiplex families that have 2 or more children. We're able to actually identify now for the first time inherited risk. And it turns out that the network modules do replicate. It's the same, that inherited risk is actually occupying the same molecular space as this de novo risk. So there really is convergence on molecular pathways during early neurodevelopment. I just bring your attention to all those blue dots up in the right-hand corner. That's the BAF complex, which is a chromatin-modifying complex involved in neurogenesis. And nearly half the genes in that 28 protein complex have been implicated already. So we start to think of autism, I start to think of it as a molecular architecture. That if we look at the kinds of mutations, red being large effect size, de novo, not under natural selection, not selected away, whereas things that are ancient and common have been pruned by natural selection, so naturally they're going to have smaller effect sizes. So when we actually look at where the large effect size mutations sit, they sit in these top tier regulators shown here, and it really fits with this kind of notion of a regulatory architecture where the synaptic genes are then being acted on through an intermediate level. So the genetic architecture parallels the regulatory architecture and really made us realize early on, and many of you may have realized it earlier, but at least for us, it highlighted the need for refined understanding of regulatory networks during neural cell lineage specification and cortical development. Just normal stuff, because knowing where to put a lot of these mutations in a functional network with regard to how the brain develops still is not known. So then what one has to do is begin to create maps. And to do that, you have to understand that there are all these hierarchies. We have the genome here. Here's a protein coding sequence in blue. And the kind of orange are all these non-coding but potentially functional elements outside of the transcriptional region. And so we have all these methods where we can identify when and where in what tissue that chromatin is open and likely being targeted using ATAC-seq. One can look at methylation patterns and histone modifications to mark enhancers. You can use evolutionary conservation. But one of the things we've learned about in human is that most regulatory interactions aren't with the closest gene. The chromatin is in a complex three-dimensional structure which brings very distant regulatory elements in contact with promoters. So we have to have also these three-dimensional maps. And since biology is driven by gene regulation that occurs at the cell type and tissue level, there really wasn't much for this in terms of fetal brain when we were starting in 2012 and 13. And it reminds me of this wonderful documentary on William Gibson, the science fiction writer who coined the term cyberspace. My disclosure is I do get royalties from this because my wife and I were involved in, in helping produce this, but that's the LA part of this, right? But the point is, it's a weird movie, but a great one. But, it's a documentary. But the point is, when I was thinking about this, it really brought, the title of this is No Maps for These Territories, and it really made me think when we were starting that there really weren't good maps, and that's what we had to do. So we've spent the last 6 years, and it's been led by Jason Stein, Hae-Jung Won, and Luis de la Torre Viedo, who now have their own labs. But we started by just taking the cortex, splitting it into the germinal zone and the postmitotic zones, very simply kind of, because we knew something about that biology, and so we had some ground truth. We did RNA sequencing to ask what genes are expressed, what regions of the genome are active, what genes do they regulate, that's the three-dimensional structure, and how do they regulate them using bioinformatics. And it's a series of papers. And so I'm just going to summarize this very briefly again, because I had to put all that genetics in the beginning. Regulatory maps help identify genes through which genetic variants are acting. And gene regulation occurs via chromosome folding that brings these genes. And so we're able to show that only half of GWAS loci are acting on the closest gene. Many are acting more than several 100,000 base pairs away, and it allowed us to kind of re-annotate the schizophrenia GWAS, which we showed in that 2016 paper. More recently, we added an ATAC-seq map, which is just looking at open chromatin regions, much smaller regions, during cortical neurogenesis, using the same tissue in the same way. And this allowed us to kind of have two independent methods to define a high-resolution map of non-coding regulatory elements that were active during neurogenesis. And then we could use those data to study human brain evolution and disease. And I'm just going to give you some examples that really gets to, just emphasize one of the points that I think, a take-home point from what Peter, our lectures, which are coming from totally different points, but I think I need to make, maybe 3 or 4 minutes just to make these points clear. If we look at the ATAC-seq points that are open in fetal brain versus adult, one can see that for an adult on, adolescent onset disease, or, one could say, when does schizophrenia start, that it's both fetal and adult risk. If we look at things like educational attainment, it's even an intracranial volume, much more so fetal. Even depressive symptoms from a GWAS is more so in fetal than adult. Autism, not powered enough at this stage. ADHD, strongly fetal. And when we divide it into the germinal zone open peaks versus the cortical plate, postmitotic neuron open peak shown on the right, we can see that most of this would be acting in the neurogenic zones during progenitor proliferation very early. And again, I'm just going to show you other lines of evidence. This just isn't one line of evidence. We have a paper on bioRxiv that defines EQTL in fetal brain. There's also Nick Bray, who's here, also had a paper published earlier last year, and this is really an important area. We hope to combine our data together, but I just want to make a couple points here. One is that the fetal E genes, that is the genes that are driven by fetal EQTL, are distinct from adult, not surprisingly. But that if we look at GWAS data and look at autism GWAS module overlap, which is shown in the yellow there, we can see that there's a highly significant overlap with fetal upper layer neurons. If we actually ask, where are the genes that are being affected by the EQTL that overlap with the autism GWAS? Again, it fits with that earlier 2013 work where the rare de novo variations. So we have evidence from common genetic variation and rare that this kind of progenitor proliferation primarily, but not only, affecting superficial layer glutamatergic neurons. And now we have splicing QTLs, and we can see that the effect sizes of these splicing QTLs in some cases is even larger than the EQTL. These are the splicing QTLs. And even though we're really underpowered for this, we can see that splicing is going to be really, really important in defining disorders, which adds another level of terrible complexity. I'm just going to go on because I'm running, I know, a little bit late. But I just felt like, especially after the discussions yesterday, I needed to just nail this point home. So here's the first cross-disorder GWAS that was done in the EyeSight Consortium. I was on sabbatical and had the pleasure to work with Thomas Warrier's group and a really brilliant postdoc, Andrew Shork, who drove this work. And so one point is, if you look up in that top corner there, this is the heritability of cross disorder. What we mean by that is I'm not calling you autism or schizophrenia or major depression. I'm just calling you affected if you have any one of those diagnoses, or unaffected if you don't. So if we compare the heritability in this same ICYC sample, which is a population-based sample in Denmark, you can see that, of course, ADHD has a very high SNP heritability, and so do the others. But the cross-disorder heritability is nearly as high, if not higher. This non-specific liability. So it's not that all liability is non-specific, that there's a component of liability that is non-specific. And one can see it here, in that if I take the top 4 SNPs in this GWAS and just show you, it's not being driven by 1 or 2 childhood disorders or adult disorders. It's really shared across them, both those that are up above the line, that are increasing risk here, and the one that is protective. And if I ask, where are the genes that are regulated by these SNPs expressed? Where does the heritability partition across the genome? Again, it peaks prenatally. This is a gene expression graph. And you can see it here, both just comparing prenatal to postnatal, 10 to the minus 30. And this is just actually showing the prenatal trajectory, right at the peak and end of cortical neurogenesis. So again, another piece of data. So lastly, in my last 1 minute and 30 seconds, I'm just going to say, now the question is, and we were discussing this a lot yesterday, and it deals with what we're going to talk about later today. How can we use all of these data to begin to inform, elucidate disease mechanisms? So one of the things, as you're seeing, we're able to kind of begin to point to cell types, developmental epochs, periods, brain region circuits. Well, then how do we study those in what organism? Well, we're beginning to develop in vitro tools, and I should say they're highly promising, but not perfect by any means. But one question we can ask is, if I have an in vivo transcriptome, methylome, ataxicome, the regulome of in vivo normal development, I can use that to ask very specifically and quantitatively, statistically, how does that map on to these in vitro systems? And we've done that several times. This was led first by Jason Stein and Luis de la Torre Ubieda in the lab, where we looked at a number of published stem cell models. And we could see that they actually do, we developed 3 tools. I'm just not going to go over them in detail, but they allow you to really quantitatively assess overlap of your in vitro system with cortical development in vivo gold standard using transcriptomics. And what they showed us at that point was that development, early fetal development, worked up to about 25 weeks, and after that, most of the culture systems at that time stopped. But Sergio Pasco has been developing, and others have been developing new systems that seem to be able to go past that and to stay more long-lived. And so I'm optimistic that we'll be soon seeing postnatal cultures, et cetera. And the point I want to make is that if I look at the modules of co-expression in vitro, I can ask, are the patterns of co-expression for autism risk genes or schizophrenia risk genes that we see in vivo, are they actually preserved in vitro? And indeed, when I look at autism risk genes, about 80% have very similar co-expression patterns, telling us that their functional regulation and likely their biological function is preserved. So I think we have systems both in vitro, and we can begin to correlate that with animal models and begin to move the field forward. And there's a lot of excitement around this, a lot of promise, but no proof. And so what we can do, though, these transcriptional profiles are giving us, we don't know what they are, right? Like as I present these, now a dozen neuronal modules that are downregulated in post-mortem autism brain. What are they? Well, we can begin to use gene knockout and connectivity maps to say this, to begin to dissect these apart. What I mean by that are there now published and online maps where people have knocked out, at Broad usually, 9,000 genes and looked at how that affects gene expression. They've also given all of the FDA-approved drugs to these cells. So we have both pharmacologic and genetic methods. And what this allows us to do now is to reverse engineer these disease networks from large-scale gene perturbation data. So we can identify drugs or gene knockouts that induce network gene expression, validate them actually in model systems, map the validated drugs back to network genes, functionally confirm using genome engineering, in other words, show that that target is indeed the critical target for the drug or the gene, and then begin to build back what are the drivers of these networks. And kind of that's what we're hoping to work on. So I'm going to end now. I'm glad that I actually thanked everybody beforehand, and I apologize for going a few minutes over. A fine-grained understanding of gene regulation during human brain development is essential to understanding the functional impact of human genetic variation, and I hope I've given you a taste of how that's really beginning to play out, that these integrative genomic and gene network approaches do provide an organizing principle for understanding molecular phenotypes in the brain and define points of potential biological convergence in disease, that autism risk genes actually do seem to converge during early fetal brain development, and this includes risk imparted by common variants, which also implicate upper layer glutamatergic neurons. Again, it doesn't mean that it's not, that interneurons aren't involved. There's quite a, it's just that if we ask for an overrepresentation of a particular neuron or system, it would be those. So genomics provides a foundation for interrogating the relevance of in vitro systems as well. Thank you for your attention.
I'm Jens Erling Leffler from the Karolinska Institute. Fantastic talk. I really love this stuff. I was curious about the enrichment that you see at early developmental stages. And you see it for autism, you see it for schizophrenia, you see it for this common shared risk. Have you done any of this, did you try to partition it? So if you remove the common risk, are these disorders, is the remaining part, is that still enriched in the early disorders?
It is. The common risk, what I meant, is entirely distinct from the shared risk between two disorders for the most part. Because the common risk, you can see it's being pushed by kind of at least 5 out of the 6 disorders for most of those SNPs. And so it's not really capturing the schizophrenia autism overlap, let's say, specifically, which is distinct from that. And so now we've been working on actually partitioning that, doing kind of local heritability mapping and partitioning the heritability locally to actually look at genes and regions, and also to look at patterns of natural selection at a regional level on these types of things. So it looks like it's, for the most part, these are distinct things, but it's telling us that disease-specific risk and the kind of shared risk, when it converges here. Doesn't mean it's not acting at all at other points. It just means that's where the strongest signal is. So again, it points to this liability. And yeah.
Hi, David Roach. Thanks, Dan. So you presented a number of sort of candidate genes that are coming out of these studies from your lab and others. I'm wondering, in a consortium like the Brain Initiative, it would be, you could imagine a filtering step would be to show the gene expression in situ, confirm at the protein level, especially if there's a sensitive critical period in a human cortical development, and that requires access to tissues. That was a bit of something we discussed yesterday. Is there an ambition within the Brain Initiative to spatially validate the expression of genes as part of the process? And can that be done sort of in tissue context as opposed to just by single cell maps? Or how will gene expression be confirmed to be at the right time and place in development to have the impact you're suggesting?
Yeah, well, it was part of the pilot projects in the Brain Initiative, but I'm not really central. Arnold might be able to answer that better, because he has a large fetal project going on now, basically, which will map the kind of cell types and gene expression during fetal development. But kind of postnatally, I'm just not sure what's really going on. Arnold, is there anything going on postnatally to the extent that you're in human?
It's a 5-year, right now, sorry. There are a few projects, and a small number that have to do with human and non-human brain development, largely fetal. We're actually the major ones working on human fetal development, and we do have, over the next 5 years, a plan to look at spatial transcriptomics. Some of that is actually in collaboration with David, you suggested or hinted at the upper cortical layer neurons and neurogenesis at the period of gestational weeks 18 to 20 or so. And it seems like the outer radioglial cells are the most likely targets for that, because they're the cells that are producing mostly the upper cortical layer neurons in human development at that age. And so it will be interesting to see exactly where those genes are expressed, and that would be an interesting candidate.
Yeah, I think validating, with the single cell work that will go on, kind of validating some of the critical genes and patterns using the spatial transcriptomics will be kind of essential to have different modalities looking at that. Absolutely.
Enjoyed your talk, Dan. Just a quick one. So you're really pointing to neurogenesis or in corticogenesis as a key factor. How much neuropathology data is there to support that in all of the brain tissues from all of these children with these disorders?
Really, I think the jury is out. I mean, when we do the art transcriptomics, it's way after stuff has happened. I think the single cell profiling will be really interesting to see if there are changes in the distribution and percentages of different cell classes, which could be consistent with an early neurodevelopmental insult.
Because one of the things we discussed yesterday in our sort of breakout groups was the problem of gaining brain material, particularly in early postnatal years. And that would seem to be fairly critical to ultimately establishing your hypothesis.
Yeah, there's one study that was published in New England Journal of Medicine that had 11 cases that showed a kind of these patchy abnormalities. And that was a little hard for me to interpret because it didn't look like a kind of known malformation of cortical development that you would, that's kind of known from model systems in humans. On the other hand, it could actually represent, something that we haven't seen before. That, so I think the jury's out. Small sample sizes, et cetera. I think the fact that there are so many, whether we look at common variation or rare variation, whether we look at an EQTL, which is a functional study, versus an open chromatin, which is a biochemical study, or a high C pattern, that we're kind of seeing this convergence, I think is more than a suggestion. I think it's quite indicative of the period. The question is kind of what? What is it affecting during that time period? We would say there's some of the genes, and because of time, I didn't show our single cell work, but it would implicate, again, there's convergence. It's mostly hitting glutamatergic neurons. Like if there's something enriched, it's glutamatergic neurons, but there are a large number of the de novo risk genes that hit interneurons and glutamatergic neurons broadly. So it's not, not every gene is specific to that thing. So there are going to be numerous routes, but it looks like this period is somewhat essential, but probably not the only unique period. It's just that we have a signal there.
Hi. So one of the things you touched on was that there's the SMPs in things like the BAF complex and the transcriptional regulators. And these would be an exception to what you said about being important during neurogenesis, because although they are important in neurogenesis, these things are ubiquitously expressed. So why do you think these would end up giving rise to these specific neurological disorders when they're expressed throughout cell types of the body?
Well, that's a really interesting question. It may have to do with, I can't speak to the extra neuronal functions of the BAF complex, and it's not every gene within the BAF complex that's been implicated. And I think it provides an interesting, that deserves mechanistic dissection. But if we look at those genes, their peak expression is around what you see there between, let's say, 12 weeks and 16 weeks fetal gestation in human brain. So it's not distinct. The particular parts of the BAF complex that are enriched, if we make an eigengene out of them, their peak expression is kind of right at the peak of neurogenesis. So it could be that what we're looking at are really those components. And I think people who are expert in that particular complex, like Jerry Crabtree, obviously you know something about this as well. I mean, it provides a really interesting avenue for studying and trying to understand that.
Certainly known that actually that complex has multiple forms with different
Yes.
components in it in different
In different tissues.
Genesis or neurons.
Yeah.
Time for coffee?
Thank you. Yeah.
Yeah. Okay, thank you very much.